Yo, what's up, Samir?
What's up, Sid? How you doing?
Doing good. How are you? What's up, movement?
Pretty good.
What's good, guys?
How you doing?
Good. How about yourself, bro?
Chilling.
Thanks for coming out.
Of course.
Thanks for having it.
Yeah. I don't know how many people are gonna show up. We got like 22, I think like 22 signups or something like that. But
Okay, that's a lot of people.
Yeah. Yeah. So, like, where are you guys like at, like, studying for that MCAT?
I'm like maybe halfway through content review ish. I don't know. I got like sidetracked at school and stuff, so I haven't been consistent.
Yeah, that makes sense.
I'm taking physics 208 right now, so I'm gonna finish that. Then I'm gonna do both biochems. Once I finish all that, then we'll start doing review.
Nice. Yeah. What's up, Cam?
Hi, Sid. How are you?
I'm good. How are you?
I'm doing well, thank you. It's been a while since I've seen you.
Yeah, it has been a while.
I think we saw you on campus recently, but that's been the last time.
Yeah. I always run into Samir on the street, but like I never see him. So like he always just like comes up to me.
Thank you for holding this. We really appreciate it.
Yeah, no
I'm
problem.
excited.
Yeah.
Very excited.
I think it should be good practice for everyone. Just learn some like concepts.
Yeah, getting into a group is motivating.
Yeah.
Being able to have a set time every week also, it's really good.
Yeah.
I agree completely. I feel like having a set time just makes it more structured.
Yeah. Yeah, I wanted also make it like as interactive as possible. So I was like wasn't like passively just like telling you guys information. Also, we got another person. Yeah, but like I was saying, I just like so like we have like a lot of questions to do stuff, like questions on this PowerPoint, questions on the passage, like besides just like the normal MCAT questions. Because like honestly, like I found like the best method to actually learn something was like to quiz yourself on it. Hey, what's up? I don't know if I can see. Let me stop sharing for a second. All right, welcome everyone. We're just like waiting for people to come in, so you go ahead and just chill for a second. All right. Okay. I think we can get started and go ahead and like start introducing ourselves, and then more people come in, then we can like talk to them. All right. So my name's Siddharth. I go to VCU. Let's go by Sid. And yeah, so today's our first session of MCAT Max, which is just kind of like this project to like provide free MCAT prep to everyone, because everyone here probably knows like MCAT prep is really expensive. It's a lot more expensive than say like the SAT, for example. Like even the official AMC products, they're all paid compared to the SAT, where you have actual free practice materials. So like what I noticed is like I took the MCAT over the summer, and I managed to score a 522 out of 528. But what I noticed is like doing well on the exam is really just learning how the AMC thinks, and also learning like they have a massive content bank that they want you to learn. And it honestly takes a lot of money to learn that like content bank. Like you have to pay for a lot of practice materials. And some people even have to pay for tutoring, for example. So I want to like provide a free resource that everyone could use to do better on the test. So like money is not really a factor. Okay. So I thought we could do like an icebreaker. So I said my name, my school. I say my favorite movie is probably like The Dark Knight, like the Batman movie. So if everyone could just go around and like say your name, your school, and then what's your favorite movie? So
My name is Kripal. I go to VCU, and my favorite movie probably have to be The Shawshank Redemption.
I've heard about that, but I never actually watched it. Like I think my roommate told me to watch that.
Yeah, it's really good. It's kind of like an inspirational story, but it's like it's really good.
Nice.
I can go next. My name is Samir Nader. I go to VCU also. My favorite movie is A Beautiful Mind.
That's the movie about the mathematician, right?
Yeah.
Yeah, that's a good movie too. So, remember that?
My name is Cameron. I also go to VCU, and I watched recently American Psycho, which I did not like because I don't like violent, heinous like things to watch, but it was recommended to me, so I watched it. But my actual favorite movie is I just had it. I forgot, but I like Inside Out, the like Disney movie. It was very sweet.
Nice.
I can go next. My name is Momin. I go to VCU as well. My favorite movie is probably The Greatest Showman. So yeah.
Nice.
All right, my name is Chelsea. I'm so sorry, my camera isn't working. But I went to UVA, and I'd have to say my favorite movie. I actually just recently watched this in theater. Woman King.
Oh yeah, I saw the trailer for that, but yeah, it looked pretty cool.
It's really good.
I don't think we can hear you.
Hey guys, my name is Ashit. I go to VCU, and my favorite movies like there's this Indian movie like on Netflix called like RRR. I don't know if any of you watched it, but like I really like that movie.
Yeah, I heard about it when it came out, like, but I never actually got to watch it. I actually want to watch it. All right, let's see. Okay. Anyone else? Okay. Okay, so today we're gonna be looking like at a biology passage from the biology/biochemistry section. So this is like honestly like I'd say one of the hardest sections on the MCAT, just because like there's a lot of stuff that they can test you on. And a lot of it is like learning how experiments work and like learning how to answer questions about experiments. So it's not just general biology, but that too, you do need to know biology to like answer the question. So we're gonna like go through some like really important concepts for the bio section, and then we're gonna like walk through a passage and see how we can tackle it. All right. So like this, since we have a small group, we can just like kind of work through all these together. So like, can anyone like here like describe this picture? You can just unmute and talk.
To me, it looks like we have a protein that is undergoing some sort of change. It looks like there was a like an OH group on it. It's being switched out for that PO3 group, PO4, whatever. And then along the way, it's somehow impacting a kinase and then a phosphatase, two other enzymes, I guess.
Yeah. So what's going on here is like, so we have like a protein, and proteins have different amino acids with different side chains. And what's really important to note is that there are only 3 side chains that can ever be phosphorylated in a protein. And that's serine, threonine, and tyrosine. So each of these are unique in that they are the only ones with the OH group. So a big theme like in biology and like cell signaling is that if you phosphorylate a protein, most of the time you're turning it on. It's not always that way, but like most of the times in the MCAT, when you have a protein, a kinase is an enzyme that phosphorylates a protein, right? So that can turn the protein on. But you can also have a protein phosphatase, which removes the phosphate, and that can turn the protein off. So like that's kind of how your cells like regulate the activity of proteins. So like, yeah. So that's something important to know. And it's important just to like pay attention to like the like PowerPoint, and then like ask any questions if you have them, because we're gonna quiz ourselves at the end to make sure like we really understood it. All right. So this is also another big concept on the MCAT is like the idea that like enzymes need other groups with them for them to work. So usually an enzyme has like cofactors and coenzymes that work with them to like help them function. So on the MCAT, there's really 2 categories of like groups that associate with enzymes, and that's called a coenzyme and a cofactor. So a coenzyme is like a non-protein organic group. So like usually when you think of organic, any compound that has carbon in it is going to be an organic compound. But an inorganic compound is most likely going to be like a metal ion, like magnesium or zinc. So a coenzyme is usually going to be organic. So like an example of a coenzyme is like you might have heard of NAD or like NADH, FADH, stuff like that. Those are all coenzymes. And so within coenzymes, there's co-substrates and prosthetic groups. So a co-substrate can bind to the enzyme, but it can also leave it, right? And then a prosthetic group, like you can kind of think like of a prosthetic leg, you always need it to be on you for like you to walk. Whereas a co-substrate, it can come to the enzyme, it can leave, but a prosthetic group always needs to be bound to the enzyme for the enzyme to work. And remember, those are both under coenzyme. So the reason we're going over this is because the MCAT will throw out like these names a lot, and like it's really easy to get cofactor, coenzyme, co-substrate, like those all sound like the same thing. So you really want to know the difference, okay? So a cofactor can be like a metal ion, like magnesium, zinc, iron. So those are all inorganic compound species. And so an enzyme that has its cofactors and coenzymes is complete, and it's called a holoenzyme. So you can think of like hollow as whole. And so an apoenzyme is an enzyme that lacks coenzymes and cofactors. All right. Let's see. All right. And we can kind of do some questions as we go along. So, okay. So kind of like our first question now is like, what enzyme adds a phosphate to a protein and what enzyme removes a phosphate? Can you guys still see the slides?
Yes.
Okay. So I guess anyone who wants to go, what enzyme adds a phosphate to a protein and what enzyme removes a phosphate?
Kinase adds and phosphatase removes.
Good, yeah. Nice job, Cam. All right. Now what we just talked about here. So like it's on the screen. So you want to try to like recall it from memory, but like since you're learning it for the first time, it's gonna be kind of hard. So what's the difference between a cofactor and a coenzyme? So someone who hasn't spoken yet.
Cofactors are inorganic, correct? Coenzymes are organic.
Yes. And then what are the two types of coenzymes and what's the difference between them?
There's a co-substrate and prosthetic groups.
Mm.
Co-substrate groups can leave or join the substrate, but the prosthetic group has to be attached like to the substrate.
Yeah, exactly. All right. Okay, so let's move on to the next one. Okay, so how many of you have taken like a biology or like physiology class where you kind of learn about like organs and stuff like that? You can just like raise your hand or yeah, it's like this is covered on the MCAT, but like a lot of people don't really learn it. So I'm just kind of like teaching a specific part of it that we need to know to like answer the passage. So does anyone know like what the kidney does? Just like a general idea.
Doesn't it filter blood?
Yeah, exactly. So basically what happens is that blood enters your kidney. I don't know if you can see my mouse, but can you see my mouse?
Yeah.
Okay, so blood enters the kidney right here. And this like circular blob thing right here is called the glomerulus. And so basically blood gets stuff in your blood, like glucose, for example, it gets pushed out into your kidney. Okay. And this process is called filtration. But what can happen is, so like I like to think of it like as you have like two like coffee cups. So you're moving a substance from one coffee cup to another. For example, you might have like glucose in one cup and then like in blood, and then you're moving it to another cup, which is your kidney ducts. So like your kidney is like a tube. So you're moving stuff from your blood into the kidney tubes, and those kidney tubes are eventually going to go to your bladder, and then like when you urinate, the urine is leaving your bladder. So so we have filtration first, and then what happens is stuff in your kidney duct can move back into your blood, and that's called reabsorption. So like you're reabsorbing that stuff from your kidney. And then also you have secretion, which is when stuff moves from your blood to the kidney. Okay. So let's see. So is it Oshri?
Yep.
All right. So what is it called when blood enters your kidney and it moves from the blood to the kidney ducts?
Is it like secretion or?
That's
To
close.
be honest, I'm not sure.
Yeah, no, that's fine. It's filtration. So you're kind of filtering the blood. So this is good to like testing yourself, like quizzing yourself is like the only way you'll actually learn it. Yeah. So even if you don't know it, it's fine. So when blood enters your kidney and it's pushed into the kidney duct, that's called filtration. So that's the first step. Okay. So also, Oshri, so like when stuff is moving through your kidney duct and it moves back into your blood, what is that called? It's on the slide too.
Is it reabsorption?
Yeah, exactly. And then lastly, when stuff from your blood moves into your kidney, what is that called?
Is that secretion?
Yeah, exactly. So we have filtration, reabsorption, and secretion. Okay. And so that's important because we also have this concept called clearance, which is like clearance kind of tells you like how well your kidney is functioning. So basically when stuff gets pushed out from your blood into the urine, that can tell you how well your kidney is functioning and also like how much blood is flowing to your kidney. So the kidney is made up of renal cells. So like whenever they say renal, that's referring to your kidney. And so if more blood flows to your kidney, more stuff is going to get pushed out of the blood into the urine, right? So if like, let's say you're a doctor and you look at a urine sample, if you like do a glucose test in your urine sample, or like a test for any other molecule, you can tell how much blood is flowing to the kidney, because if there's more stuff in the urine, that means more stuff is being pushed out from the blood into the kidney. Okay. But the thing is, we can't really tell blood flow to the kidney if stuff is getting reabsorbed from the urine to the blood, right? How can we tell if the kidney is really working if anytime we push something out, it just goes back into the blood? So that's why if you do a urine test and you look at and you're looking at a substance to tell how well the kidney is working, you want a substance that's not going to be reabsorbed a lot. You mostly want it to be filtered and secreted, so so it actually stays in the urine. So when you look at the urine, you can tell how well the kidney is actually functioning. Okay. So can anyone just like summarize like why you want a substance that is filtered and not reabsorbed to like tell how well the kidney is functioning?
I can do that.
Yeah.
So since through filtration, the blood is going into the kidney, and then through reabsorption, it's going back into the blood. We want something that doesn't go back in the blood so that we can have a good marker of how good the filtration of the kidney is going.
Yeah, exactly. And then also just like, remember like the actual blood is not moving into the kidney ducts. It's like the stuff within the blood, but like you got everything else. Yeah. Right. So awesome. This is honestly like a tough concept to understand. It's kind of like unique, like it's like not tested that much, but it's like, it's so important. Okay. And okay. So these next few slides are really, really important for the MCAT. So these are like biochem technique, like biology/biochem lab techniques to like separate proteins from each other. Okay. So the biggest one is called affinity chromatography. So can someone explain what a ligand is? It's like just like a simple sentence, or like
It's like any molecule.
Yeah, any molecule that binds a protein is called a ligand. So we can actually use this to our advantage. So let's say you're working in like a biology lab. You have a bunch of proteins that are mixed together, but you want to separate a protein from all the other proteins. So how that's going to work is that you're going to use the fact that a protein binds its ligand to separate it from all the other proteins. So what we have right here is we have like a tube, and we have like beads inside the tube. So what we do is we attach the ligand to the beads, so that the ligand can bind the protein. So let's say you pour all the proteins through here, the ligand on the beads is going to bind the protein. So the proteins stick to the ligands and everything else just like washes through. All right. And so, okay, now we finally have our proteins stuck to the ligands on the beads, but we want to kick our proteins off. So what we do is we use the salt to kick it off. And that's how we like isolate our proteins. All right. So could someone just like describe that process for me? Chelsea, are you still there? Someone else, maybe. So like anyone can go. Just like give it your best shot.
I'll go.
Yeah.
So it looks like we are manipulating the fact that a ligand will bind to a protein in order to separate it out in solution.
Yeah.
So we find that there are other components in the mixture that do not bind the ligand. So we can put the ligand into the solution and they will bind to the proteins. And once that it is bound, the other components of the mixture can be washed out. And in order to kind of, now that we have the proteins isolated, we can use a salt to stick the ligand from the protein so that we're left with just the protein in the solution.
Awesome. Yeah, that's exactly how it goes. Yeah, great job. All right. Yeah, so that's one form of protein purification. Okay. Okay, now we have two really important techniques. So this is called a northern blot and a southern blot. So a northern blot is when we have a bunch of RNA, but we want to separate it. So what we do is so we have a bunch of mixture of RNA fragments, and we use something called gel electrophoresis, which separates them by their size. Okay. So once we separate it by their size, we move all the RNA to a blotting membrane. Okay. And once it's on the blotting membrane, we use a probe. So a probe is something that's complementary to the RNA, and it binds to the RNA so you can detect the RNA. Right. So this allows us to detect, let's say you have a cell, but you want to tell whether it expresses a certain gene. So you want to tell if it makes RNA of that gene. You can break up the cells and look through the RNA, separate them, and use a northern blot to detect whether certain RNA is within that cell. So this is very experimental, and that's just the nature of the MCAT. It's a bunch of experimental techniques. Okay, and now we have a southern blot. So a southern blot is the exact same thing as a northern blot, except you just have DNA. So you use, again, a gel electrophoresis to separate all the DNA fragments based on their size. So bigger fragments are going to move slower through the gel, whereas faster fragments are going to move, smaller fragments are going to move faster. So once you have that separation of the DNA, you transfer it to a blotting membrane. And again, you use a radioactive probe to detect the DNA in your sample. So you're looking for a specific DNA in your sample, just like you're looking for a specific RNA in your northern blot. So could someone describe northern blot / southern blot to me?
Yeah, so with northern blot, you have a lot of RNA, and you want to isolate the RNA, so you use gel electrophoresis to separate the RNA molecules on size, and then you transfer them to the blot. And then on the blot, you have RNA probes that will help you label and determine which RNA you want to analyze. And then southern blot is essentially the same thing, except you have DNA instead of RNA.
Yeah, exactly. Yeah, you guys are doing really great. You're picking it up really fast. All right. Yeah, so that's exactly how northern blot, southern blot works. Oops. All right. Now we have one of our last, probably, yeah, it's one of the last techniques for protein purification, and that's ion exchange chromatography. All right. So here we utilize, remember in affinity chromatography, we looked at protein ligand interactions to separate proteins. In this case, we're using the charges of proteins to separate proteins. So we have this is called a cation exchange column because it binds positive charges. So we have negatively charged beads, and those beads are going to attract positive proteins. So let's say you have a mixture of proteins. The positive proteins are going to bind to the beads. The negative proteins are going to repel the beads. So you're going to have the positive proteins sticking to the beads, right? And then once you wash off all those other negative proteins, you have your positive protein that you want on the beads. So you need to kick it off. And just like how we did in affinity chromatography, we use a salt to kick it off, and that's called elution. So could someone describe this process to me? Ashrith, could you go ahead and give it a shot? Just
Yeah, so
Mhm.
yeah, ion exchange chromatography is a method to separate proteins based on their charge. So for cation exchanges, all the cations will stick to will bind to the negatively charged beads within the column, and all the negatively charged proteins will elute off the column because they're repelled by the negatively charged beads. So therefore you separated the proteins based on charge. So you have all your negatives eluted off, and you have your positives still in the column.
Yeah. Awesome. Yeah. That's great. That's how it works.
I have a question. Why wouldn't the positive and negative, or the cation and the anions in solution, just stick to each other? What makes it want to stick to the anions on the surface of the container and not to the other negatively charged proteins?
That's honestly a good question. I think part of it is that like the anions are stuck to the tube, whereas like gravity is acting on all the other negative anions. So you can imagine those negative anions are like falling, whereas those other negative anions are like stuck to the thing.
Catching almost. Okay.
Yeah, exactly like that. That's how I think of it. And then they probably have just like they try to put more negative beads in there than there are negative ions. So like it's like competition kind of. So that's yeah, that is a really good question though. I'm pretty sure, yeah, that's just how I think about it. But if you Google it, there could always be like some other explanation. But yeah. Yeah, but
thank you.
Yeah. That was a really good question though. All right. Lastly here, this is something you just like kind of have to like memorize. It's like these are all the types of enzymes. So they want you to know all the names of the enzymes. So we have our oxidoreductase. That's kind of like, if you're familiar with oxidation reduction reactions, we like transfer electrons. So based on the number of electrons you have, that determines something called an oxidation state. And so an example of that is called a dehydrogenase. Then we have a transferase. You transfer a functional group from one molecule to another. So remember we talked about our kinase, which like phosphorylates a protein. So that is an example of a transferase. We have a hydrolase, which is like a lot of times you have a protease, which breaks down proteins. So proteins have peptide bonds, and you use water to break down those covalent bonds. So that's a hydrolase. A lyase, you break down a covalent bond, but you don't use water. And a decarboxylase is an example of that. So an isomerase is when you just switch up the bonds within a molecule. So you don't actually change the molecule, you just like switch up how it's connected. And then a ligase forms a covalent bond between two large molecules. So this chart you just like need to memorize. Like, yeah, it's just something you need to memorize. And like, if you can know the names of the examples, you might like, you'll definitely get a question like, what's this type of enzyme called? And you don't want to like lose points on that. All right, so we're almost at the end of our PowerPoint, and then we'll actually get to the passage. Okay, this is another like big biology concept. So does anyone here know the differences between the three types of muscles? And like, it says it on there, but like, yeah, if anyone wants to just go ahead and like, if they know it.
So I don't really know the difference between like the striations and stuff, but I do know skeletal muscle is everything like attached to your like skeletal system, like muscle wise. Cardiac muscles, anything with like the cardiovascular system, and smooth muscles are like your like digestive system, things like that.
That's right. Yeah, so skeletal muscle, the big thing with skeletal muscle is that's like it's muscle you use like voluntarily. So like you can, when you walk, you're using skeletal muscle. When you hit the gym, you're using skeletal muscle. But smooth muscle is something you really don't have any control over. It's like controlled by your autonomic nervous system. It's involuntary. So like you can kind of think like you can't consciously control the muscles in your stomach, right? They just kind of like work on their own. So that contraction is involuntary, and that is smooth muscle. Okay. And so also another big feature, they'll like test you about this random fact, cardiac and skeletal muscle, they have stripes on them. And so those stripes are myosin and actin binding to each other. smooth muscle doesn't have that type of structure. They have myosin and actin, but they don't have the stripes that cardiac and skeletal muscle do. And also, the last fact that you need to know is that skeletal muscle has a bunch of nuclei within one cell, but skeletal muscle is actually made up of a bunch of cells that have fused together. And so when cells fuse together, your nuclei combine into one cell. So that's why you have a ton of nuclei in one cell. And then cardiac and smooth muscle, they both have just like one nuclei in their cell. So those are the big differences. And lastly, there's this concept called peristalsis. Does anyone know what peristalsis is? You can kind of guess just based on the picture, but like it might not be that clear. All right. So peristalsis is basically like the automatic contraction of the smooth muscles in your GI tract. So the muscles in your esophagus, when there's food traveling down it and through your stomach, they just automatically contract like rhythmically. So like one contracts, one relaxes, then the other one contracts to like push food down your stomach. So that's called peristalsis, and it's controlled by smooth muscle. Okay. So that's an important concept to know. All right. All right. Let's see. Okay, this will be good exercise. So can someone go and try to name as many classes of enzymes you can from your memory? Even if it's just like 2 or 1.
I remember hydrolase and
Yep.
lyase.
Yep.
And no, I'll just.
Yeah, that's good.
There's transferases, ligases, reductase. Yeah, that's all I can think of off the top of. Oh, isomerases. I don't know if I said that already.
Yeah, I think we covered all of them. Let me see. Hydrolase, isomerase, ligase. Yeah, I think we covered all of them. That's great. Yeah. And so lyase is like, whenever I see lyase, I don't really know. When I was studying, I didn't really know what it meant. But like, basically, it's just like when you remove something from a molecule, like that you don't use water, which is like, how do you know whether something uses water or not? But like, anytime you remove a CO2 molecule, a decarboxylase, you know that's a lyase. So that's kind of just like a small esoteric fact that you just have to know. Okay. Yeah. So that was the enzyme classes. And then let's see. So can someone tell me what are the differences between smooth and skeletal muscle?
The biggest difference that I know is that skeletal muscle is voluntary movement and smooth muscle is involuntary, used by your autonomic nervous system.
Yeah, exactly. All right. And is peristalsis example of smooth muscle contracting, or is it skeletal muscle?
Smooth. Smooth.
Oh, sorry, who's someone calling me? Oh, so did someone say smooth?
Yeah, I said smooth.
Oh, so okay, sorry, I didn't hear you. Yeah, that's right. All right. Okay. Yep. So that was basically the background intro that we needed before we could actually look at the passage. So now we're going to actually look at the passage. All right, let me share my screen. All right. So can everyone see the screen? Awesome. So these are like some Jack Westin questions. So if you go to jackwestin.com, you can find free MCAT questions, but like thing is they can be kind of hard if you don't know how to walk through them. So we're going to walk through a passage and actually try applying all the content we know, because anyone can just like learn all the concepts they need to know for the MCAT, but applying them is another skill in of itself. So
Quick question.
yeah.
How similar is Jack Westin to the actual like MCAT in terms of like content?
It is pretty similar, I would say. At times they can even like copy like AMC questions, like they straight from the practice. They like maybe switch up a thing or two, but it's like copied from the practice.
Gotcha. Appreciate it.
Yeah, no problem. Okay, so how I'm thinking is like if someone could read this first paragraph for me, that'd be great.
Yeah, I'll read it.
All right.
Serotonin is a monoamine neurotransmitter derived biochemically from L-tryptophan, figure 1. The release of serotonin from enterochromaffin cells in the digestive system has several essential functions, including the initiation of peristalsis. The rate-limiting step in the synthesis of serotonin in enterochromaffin cells is the hydroxylation of L-tryptophan. The rate of this reaction is influenced by tryptophan concentration, the calcium-dependent phosphorylation of the enzyme, and cofactor availability. Serotonin can be metabolized in the kidney and liver by monoamine oxidase and aldehyde dehydrogenase to 5-hydroxyindole acetic acid, which is secreted into the urine. 5-HIAA has a high ratio of filtration/secretion to reabsorption. The serotonin receptor family is a large family of G protein coupled receptors and one ligand gated ion channel, 5-HT3, which is a non-selective cation channel. The receptors are present on neurons in both the central and peripheral nervous system. The major excretory metabolite of serotonin, 5-HIAA, has been proposed as an indicator of brain serotoninogenic activity over the winter. Low levels of 5-HIAA correlate with the incidence of seasonal affective disorder, SAD.
All right, thank you. So just based off that, how are you guys feeling about the passage? Does it seem hard or yeah? Some of the jargon can be like pretty confusing, especially when you first look at these passages. Like that's how I felt when I first looked at like biology passages, like they were pretty confusing. So I think the best thing that helped me with studying was like quizzing myself on the passages, like besides just like the questions on the passage, like coming up with my own questions, like comprehension questions, to actually make sure I understood the passage. So that's what we're kind of do right now. So I have some questions and I'll just read them out. Okay. So what's the main molecule being talked about in the passage?
Serotonin.
Awesome. So what does serotonin do? What's its function?
It's a neurotransmitter.
Yep. And what can it kind of like do in the digestive system? Like how does it relate there?
It initiates peristalsis.
Awesome. And what's peristalsis?
The automatic contraction of smooth muscles
Yeah.
to like push food down.
Yeah, exactly. So what is serotonin made from?
Tryptophan.
Yeah. So this is L-tryptophan. And what is tryptophan? Like, what do we call this thing?
It's an amino acid.
Amino acid.
Yeah, exactly.
Precursor.
Yeah. Yeah, so it's an amino acid, a precursor. And what factors influence how fast the serotonin is made?
Its synthesis is the rate limiting step, which relies on a number of precursor things. So it relies on the calcium dependent phosphorylation of the enzyme, tryptophan concentration, and availability of cofactors.
Right. So remember, what's a cofactor? We talked about in the PowerPoint, but does anyone remember?
Cofactors are the inorganic things that help enzymes work.
Right. And so what is the enzyme doing in this case? Like, what is it doing to tryptophan? It says it in the passage.
Hydroxylating it.
Exactly, yeah. So what the passage is looking at is like what affects the rate that the enzyme adds that OH group, the hydroxyl group, to tryptophan to make serotonin. So like Cam said, it depends on the tryptophan concentration, right? Because if we have more of our starting material, more of that tryptophan, we can make more serotonin, right? So anytime you have more of a substrate for an enzyme, the reaction rate increases. And then remember how we talked about a kinase and phosphorylation? So we talked about how phosphorylation, adding a phosphate group to an enzyme, can turn enzymes on. So just like we talked about that, phosphorylating this enzyme can increase the rate that it makes serotonin from the tryptophan, that it hydroxylates this tryptophan. And if you have more cofactor, remember cofactors help the enzyme with its function. If you have more cofactor, you can hydroxylate the L-tryptophan faster.
Wait, can you repeat that? Did you say that that more Ca2 means more phosphorylation, or is it the rate of the phosphorylation which is increasing?
Yeah, so basically, so when you phosphorylate the enzyme, you're increasing the rate that the enzyme can hydroxylate the tryptophan. So the phosphorylation of the enzyme and the hydroxylation of tryptophan are different. So like when you phosphorylate the enzyme, it can do its job better, like it's activated to hydroxylate the tryptophan, which will eventually lead to more serotonin because serotonin comes from tryptophan.
So you're saying the more Ca2, then the more phosphorylation which can happen, which means the faster that serotonin can be produced.
Exactly. Yeah.
All right.
And same thing here, like with cofactor availability. If we have more cofactor, it can bind to the enzyme and help the enzyme hydroxylate the tryptophan. So what you want to do in your mind is anytime you're reading a biology passage or any passage on the MCAT, you want to kind of have like a mental framework of like what's being talked about in the passage and how things relate to each other. Right? It's going to make the questions a lot easier to answer. So right now we're taking time to go through the passage and review it and like quiz ourselves on it. And that's really important because like soon you'll be able to do these things like subconsciously when you're reading the passages, but like you'll be able to breeze through them. So they become a lot easier for you to read. All right. So when we have enough serotonin in the body, what does the body do to it to like kind of get rid of it?
Breaks it down.
Yeah. So where is it broken down?
Kidney and liver.
Right. And what enzymes do that?
MAO and the aldehyde dehydrogenase.
Exactly. And so what is that metabolite? So metabolite is usually when you break down a compound, you call it a metabolite. So what is that metabolite called?
5-HIAA.
Exactly, yeah. And then so that 5-HIAA is secreted into the urine. So once the liver and kidney, they break that down, the serotonin down to 5-HIAA, you push it from the blood into the urine in the kidney. And then once it's in the urine, it leaves your body, right? So it says 5-HIAA has a high ratio of filtration/secretion to reabsorption. Why could that be significant? Like based on what we talked about in the PowerPoint, why could that matter? Like, why is that important?
We can actually use a urine test to figure out if the kidney and liver, or at least the kidney, is breaking this down.
Yeah, exactly. So like whether the kidney is like filtering it out of the urine, like whether the kidney is properly functioning, it's filtering blood, right? Because you want something that's actually going to be filtered and not pushed back into the bloodstream. So when it comes out in the urine, you can look at it in the urine and you can determine whether the kidney is actually filtering the blood. Awesome. Okay. And then here we talk about the serotonin receptor. So can someone tell me about like what the serotonin receptors are?
So it's a G protein coupled receptor. I think that's metabotropic. There's an ligand, and then there's another one. It's also a ligand. No, it's a ligand gated ion channel. 5-HT3.
Awesome. So where are these receptors usually?
In the postsynaptic cell from where it's being released.
Yep. So like neurons throughout the nervous system, exactly, postsynaptic. So then here at the end, they tie in like a random like disorder. So like that, a lot of times they'll like throw in like a name of like a random disease or like a medical condition, and they'll tie it back to whatever you're talking about in the passage. So the thing is, you don't really need to know anything about that random thing that they throw out at you. You just need to know how it ties in to like 5-HIAA and serotonin. So can anyone explain, like just based on your hunch, why 5-HIAA, why low levels of that could be related to seasonal affective disorder? Just like,
Well, I know that like serotonin as a neurotransmitter controls a lot of the like feelings and subjective kind of like happiness that we feel. And if we're able to test urine for a low level of 5-HIAA, means there also might be low levels of serotonin in the brain. That could be a deficiency that's leading to a disorder like SAD.
yeah, that was exactly my thinking too. Yeah, so I'm pretty sure that's right. Yeah, awesome. So now that we have a good understanding of the passage, it's gonna be a lot easier to answer these questions. So here they have a diagram just kind of showing how you make serotonin from tryptophan. And you can see right here, they add no age group right here. All right. Awesome. So can everyone see question 1? Okay. So can someone read question 1 for me?
Based on the passage, which of the following within enterochromaffin cells is not likely to contribute to SAD? A, decreased tryptophan levels. B, decreased oxygen tetrahydrobiopyrin levels. I did not say that correctly. C, decreased phosphatase activity, and D, decreased intracellular phosphate levels.
Awesome. So this is kind of forcing you to apply the knowledge. Does anyone have any idea of how to tackle this question?
I don't know. What someone?
I kind of have like a strategy, I guess. I see each of these answers is kind of targeting like a different thing. So for example, like tryptophan levels in A, and then C is talking about phosphatase activity. What I would do is I would go through each one and try to like think about, okay, we have tryptophan. How does that relate to serotonin and SAD? And then if we were to increase tryptophan, what would happen? If we were to decrease tryptophan, what would happen? And then the same thing for phosphatase, and then each one, and then come to an answer by doing that.
Yeah, that's exactly how I would tackle it. You would have to go choice by choice and see how it affects serotonin levels. So this question is really testing you on how changing each of the steps in the pathway affects serotonin levels. So first, let's talk about SAD, like seasonal affective disorder. Do we have low or high serotonin levels in SAD?
Low.
Low, exactly. So we want to figure. So the question says, which does not contribute to SAD? So we want to figure out which choice is not going to cause low serotonin levels. Okay. So we know serotonin is made from tryptophan, right? So is choice A correct or is it wrong? Sorry, I didn't hear you.
I said A is incorrect.
Exactly. And why is it incorrect?
Because tryptophan is a precursor to produce serotonin. So if we decrease that precursor, it's a rate limiting reagent. So it would not.
Yeah. So yeah, it would contribute to SAD. So B. So if you look at this diagram, so you probably don't know what O2 tetrahydrobiopyrin is. So, but if you see here in this diagram, it's kind of used it's used in the pathway to make serotonin, right? So if we have decreased amounts of this compound, what's going to happen?
Less serotonin produced.
Yes. So does that mean the answer is correct or wrong?
It's incorrect.
Right. Because it will contribute to SAD. All right. So if we have decreased phosphatase activity, how's that going to affect serotonin levels?
If we increase the phosphatase activity, then that's going to increase the speed at which serotonin can be produced. So if we decrease phosphatase activity, then that would slow down how much serotonin will be produced. So I think that would contribute to SAD. So it's incorrect.
Right. So let me think of it. So if you decrease the phosphatase activity, you will leave the phosphate group on the enzyme that helps make serotonin.
Oh, phosphatase is taking off the phosphate group. Oh, okay.
So if you decrease the phosphatase activity, you're removing the phosphate group off the enzyme less. So the phosphate group stays on the enzyme. Remember, it says here, the rate of the reaction is influenced by phosphorylation of the enzyme. So if you leave the phosphate group on the enzyme, the reaction is going to go faster, right? So it is right that this is not going to contribute to SAD, right? Because if you leave the phosphate group on, the reaction goes faster, you make more serotonin. So it's not going to contribute to SAD. So that is right. So yeah, so that one's kind of complex just because there's a bunch of moving parts in there. So you kind of have to like think it through. So does everyone understand why C is correct? Everyone? All right.
But so one more time, you said that phosphatase takes off the phosphate group.
Yeah.
So if we have lower levels of the thing which is speeding up the serotonin production,
So
then there's going to be more serotonin.
So we have lower levels of the enzyme that is taking. So we have an enzyme that makes that helps make serotonin from tryptophan.
Yeah.
How fast that enzyme operates depends on whether it's phosphorylated or not. So if we decrease the activity of the phosphatase, we're leaving that phosphate group on the enzyme, so that enzyme is going to work faster. So when that enzyme works faster, you make more serotonin. So the phosphatase is not affecting the serotonin; it's affecting the enzyme that makes the serotonin. Right? Okay. And then D. Why is D incorrect? We could just use process of elimination, but why is it incorrect?
Since the phosphorylation of the enzyme is part of the precursor to produce serotonin, if we decrease the phosphorylation level, the intracellular phosphate levels, we're not going to be able to do one of the precursors to produce serotonin, and this is going to decrease the amount of serotonin, which would contribute to SAD.
Yeah, that's right. But remember, the phosphorylation is happening to the enzyme that makes serotonin. It's not actually happening. It's not actually phosphorylating the precursor itself. So that's just like a detail that you want to pay attention to. But yeah, it's the end result is the same. All right, so the answer is C. You can see we got it right. So they have answer explanations on here, but like you can see they're like not very extensive. So if you don't like really know the concepts, like it's going to be kind of tough to understand them. But if you ever want to do practice on your own, I think they could be helpful. All right. Awesome. So could someone read question 2 for me? See if Chelsea's here. Chelsea, are you there? No. Oshirth, could you read question 2?
Yeah, so the question 2 is, which of the following is true regarding 5-HIAA?
Awesome. So this is going back to the kidney stuff that we covered in the PowerPoint and reabsorption, filtration, all of that. So does anyone have an idea of what the answer is?
I believe it's A.
Yep, exactly. So could you explain how you got that answer?
So I kind of looked at like the whole thing in like the passage where it said 5-HIAA has a high ratio of filtration / secretion to reabsorption, which means that the majority, so not a lot of like the particles that are in the blood or that go through the kidney, they don't go back into the blood. So it's a good indicator in like a urine test, for example.
Exactly. Yeah. Because it says right there, you have a high ratio of filtration to reabsorption. You can tell how much stuff is being pushed into the urine from the blood and the kidney because it's not being reabsorbed. So it's going to stay in the urine. So it can kind of tell you about how good the kidney is functioning. So does that make sense to everyone? Anyone have any questions? Always like, if you have any questions. Oh yeah.
Is renal clearance only applicable to secretion, or is there a different like kind of terminology for things that are filtered through reabsorption?
So clearance refers to, it's like filtration, reabsorption, and secretion like combined. So how you kind of look at it is filtration and secretion are both pushing stuff from the blood into the urine, whereas reabsorption is moving stuff from the urine back to the blood. So they're doing opposite things, right? So you can kind of take your filtration and you subtract it from, you subtract whatever you're reabsorbing from whatever you're filtering and secreting, because it's moving in the opposite direction. So if you have more filtration than reabsorption, more stuff is going to be in the urine, right? But you have more reabsorption than filtration, more stuff is going to be moving back to the blood. So it's like it's like a tug of war, basically. So yeah, so clearance is kind of like the net result of all that. So basically, which one wins, reabsorption or filtration? So, yep. Yep, and it's right. Awesome. Can someone read question 3?
Which of the following laboratory techniques would most likely be used to measure the association between tryptophan hydroxylase and its cofactor?
Awesome. So does anyone have an idea what we could use?
Just going through A and B, I know that those are kind of using trying to like RNA and DNA, and we're talking about enzyme and a cofactor. So I really don't know if those would be very like useful. So then I would look at affinity chromatography and ion change chromatography. The ion exchange was one that utilized the charges of each, and I can't remember if we went directly over affinity chromatography. So I'm thinking C and D.
C is correct. So affinity chromatography is the correct answer because association between an enzyme and its cofactor is kind of like, it's like you're measuring their affinity to each other. So if you attach like the cofactor, let's say, to a bunch of beads, and then you put the protein mixture in, the enzyme is going to bind to the cofactor. So if you isolate a bunch of enzyme, that means the enzyme bound to the cofactor really strongly, right? So it was able to be separated. So you can measure its affinity, its association. But if it doesn't get isolated, then it just acted like all the other proteins. It didn't really have any high affinity for the cofactor. So does everyone get why it would be affinity chromatography? All right. Awesome. Let's see is the correct answer.
I have a question.
Yeah.
Would a cofactor be considered a ligand then in that case?
Yeah.
Since it's okay.
It would be a type of ligand.
Okay.
Samir, did you have a question?
No, I was just wondering, like, in what case would ionic, like, what would have had to be different about the question for D to have been the right answer?
So if they mention anything about like charge or anything like that,
Okay.
like let's say the protein is negatively charged, or there's something called an isoelectric point, which is a point when a protein is neutral. So sometimes a protein will have a charge. So if they mention anything about isoelectric point, which we're not going to cover today, but if they mention anything about the charge of the protein, then it would probably be ion exchange chromatography. But
Sure.
because said association between an enzyme and its cofactor, it's probably going to be affinity chromatography. Okay, awesome. Almost done. All right. Because someone read question 4. I need to use the chart to answer this.
The enzyme which catalyzes the conversion of 5-hydroxy-L-tryptophan to serotonin is most likely classified as which of the following? A. Oxoreductase, B. Hydrogelase, C. Lyase, D. Isomerase.
Awesome. So that's looking at this one. So what's the difference between these two molecules? Sorry. Can anyone spot the difference?
The first one has a carboxylic acid group, and then it's replaced by, I'm trying to remember, an amine group, I think.
Yeah, basically like you're right, the carboxyl group is just removed. So what enzyme removes the carboxyl group?
It would be a lyase.
Yes, exactly. So that's how we answer this question. All right. Lyase. That's right. Okay, last question of the day. So can someone read question 5, the last question?
In the digestive system, serotonin released from enterochromaffin cells is most likely responsible for which of the following? A. The contraction of skeletal muscle. B. The inhibition of skeletal muscle. C. The contraction of smooth muscle. Or D. The inhibition of smooth muscle.
Right. So what is everyone thinking? So what is
C.
Yep, exactly C, because it's responsible for initiating peristalsis. Peristalsis is contraction of smooth muscle. Awesome. And that is the end of the passage. Let me stop sharing my screen. All right. Thank you guys so much for coming out. Hopefully next time we'll have some more people. So like, if you know anyone, like try to like recruit them. Like if anyone might need like MCAT prep, but that was it for today. And thank you so much for coming. And I should set a date for another session. It'll be next Monday or the next Monday after. We're still figuring out the schedule, but yeah, today was really good, and I hope you learned something from it. Yeah. So what was your general feedback on the session? Like, what would you improve, or like, what do you like about it?
I think you did a great job. I feel like it was very educating. I like the structure of like explaining like all the background first, then us moving towards the passage. I think that was a really good idea. But yeah, I don't have much like negative criticism. So I think you're doing good.
Thank you.
I also really like the active recall, like answering questions after you did the explanation, like after you did the background information, the active recall, I think that really helped.
Yeah. Yeah. Thank you so much. Yeah. Like I would say, honestly, the biggest key on the MCAT is like active recall, quizzing yourself. I honestly that's like honestly a secret that like a lot of people don't talk about. But yeah, anyone else?
Honestly, good job, Seth. I really enjoyed it. You did a great job. You should be
All right.
proud.
All right. Thank you, Cam. Thank you so much.
Thank you so much. I learned a lot, and it was amazing. If I could give one suggestion that would maybe help? I like to think through things like very logically, like step by step. And that first question was really like it was like a negative. Also, it was like which then would not contribute to SAT, which was the lack of serotonin. It was like a lot of like double negatives going on. So if you like after the question was finished and we had like the answer, if you could have like switched to like a picture of like the exact pathway and like what was being influenced that caused that and why that was our answer, that would have like been amazing. Just like see it all come together. I had to like think about it while we were on like the second question for like 5 minutes still after trying to think about the first one. And then it finally clicked. I was like, okay, you know what I mean? Like the whole pathway and how it affected it. So maybe if you had just that like exact like boom, 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 and that's why like, you know what I mean? That would be like amazing.
Yeah, I think, that definitely sounds like it would be helpful. So I'll definitely work on something like that. If we have like a similar question in another.
Yeah. Thank you so much. Though.
Yeah, no problem. I think Ashrith, were you trying to speak before?
Oh yeah, I mean, I echo a lot of the feedback that other people have been giving, but I have a question. So like, will we get this PowerPoint to us, emailed to us at any point?
Right. So I'm gonna upload the PowerPoint and the questions on the website, and also a record. So a recording of this video, so like anyone can watch it if they want to like see what we went over. Just like so we have a resource like on the internet to help people. So and like I would really appreciate if you could pass it on to anyone else. Like we had a lot of people sign up, but I guess like people got busy or just forgot. So just trying to get more people involved. But yeah. Yeah, so thank you so much. And I think everyone's good to go.
Thank you.
Awesome. Thank
Thank
you.
you.
All right, see
Thank
you.
you.
Thank you, Sid.
Yeah, no problem.